We can now take cells from those patients and we can generate stem cell lines unique to that patient. We can expand them. We can turn those cells into the organs, so liver cells or kidney cells. So the next step would be then putting all these together with the patient's own cells and creating a new kidney for them, which, as you know, there's a huge demand, a huge shortage
Yeah,
of
yeah.
organs
Mhm.
for transplantation. So that, I think, is I think it's unfair to call it the moonshot, probably more like the Mars shot, but that is what
Yeah.
we all work towards. And that is the goal that brought me to Cellbricks. And that is what we mean by tissue therapeutics. It is a therapeutic piece of tissue or eventually organ that would be transplanted or implanted into the patient to recover loss.
So hello, Simon, and welcome to Impulse. I have to say I've been, looking forward to this episode for a while now, even actually before we were in touch. So, for those who know me, I studied biomedical engineering at EPFL, and the specialty I took during my master's degree was regenerative medicine. So it encompassed subjects like biomaterials, stem cell engineering, and we had different research projects where we were trying to combine synthetic materials, sometimes 3D printed, with living cells and tissues. And I still find it's one of the most exciting fields of research in medicine. And although I've been kind of like away from it for some years now, I've been wanting to cover the topic on the podcast since the beginning, to understand the most recent progress we've made in this space. And to be fully transparent, I was in touch with someone you probably know, called Eric Gattenholm. He was the founder of Cell Inc., which then became Pico. And we had agreed on the recording date a few years back, and we unfortunately had to cancel, close to the agreed date. And we actually didn't manage to find another time until he stepped down of the company in 2024. So, all of this to say that I'm really glad that, I came across your work there that you're doing with Cellbricks. And I'm delighted that, having someone like you on the show with whom to dive into the world of bioprinting and the fabrication of living tissue. But before we dive into these topics, and for those who might not be familiar with you just yet, I would simply ask you, Simon, to present yourself.
Absolutely. Thank you, Matthew. Um, so my name is Simon Mackenzie. I'm a biochemist by training. I've worked in many different areas of life science, from drug discovery with small molecules to proteins to setting up service businesses. But where I've really focused most of my effort has been in trying to help young companies develop and grow, and particularly in the space that we're going to talk about today. And that's encompassed building companies that are in cell therapy, stem cell companies, biomaterial companies, and also companies that build bioprinters. And bringing all those together brought me to Cellbricks.
And what drew you initially to this field of like tissue engineering and this, because it's quite a specific, let's say, part of the, I would say, medtech business.
Yeah, I think it's very interesting you brought up Eric. Now, bioprinting or 3D printing. So 3D printing in biology had been around for quite some time, but I don't think it really identified what its niche was. And yes, people could say, absolutely, one day we will print organs, and that's a great goal. But in reality, it's like trying to put someone on Mars, the number of different technologies you have
Yeah.
to bring together. And most of those technologies, until very recently, just didn't exist. So there was really a limit to what you could do. So I was involved in a lot of the enabling technologies being built. So stem cells. I worked in a number of companies where we've started off with embryonic stem cells and then went to iPSCs. And
Yeah.
that really was for building disease models. So the idea being creating microtissues,
Yeah.
something, even just 2D to start with, something that gave you something more physiologically relevant than a standard 2D cell model that might allow your therapeutic program to be able to be predicted better for success in the clinic. And then, of course, 2D wasn't enough, even with these expensive cells. But the problem was that the whole process was incredibly expensive. So it was only used at very late stages. And for a lot of people, it just wasn't possible to do that in terms of finance or access. So I could see that stem cells have come a long way and now are such a routine tool that everybody uses them. Um, but then a lot of these other technologies, you also mentioned biomaterials. Again,
Yeah.
biomaterials have been around for quite some time. But as a tool, as a research tool, absolutely, people can try and formulate new things. But when you actually then move it to start to become a product, it's a very different mindset and a very different set of science that needs to be involved. So biomaterials starting to come together, cells, I was building these complex cell models. I was then lucky enough to move to Switzerland, where I helped develop a bioprinting company. So this was an engineering company. All we focused on was the hardware and the software. And
Yeah.
there, my role really was to try to understand what was the market for it. And I come back to Eric. So Eric with Cellink to start with, I think was in the first wave of bioprinting. And this is
Yeah.
where everyone wanted to try and do bioprinting. And he was able to democratize. He was able to, um, I guess, the main mover for this access to these low cost or lower cost bioprinting systems. Unfortunately, I think what people discovered is it's a very complex thing. So there was
Yeah.
lots of interesting research, but really didn't move to application.
Hmm.
So when I came across Cellbricks, what I felt was the pieces of the puzzle to do tissue therapeutics actually potentially existed. And Cellbricks had the pieces of the puzzle. They maybe hadn't focused or they weren't using them quite all in the right manner. But
Hmm.
I felt that was possible. And the board and the senior team at the time said this was their ambition. And I thought, someone's got to do this. And having been in all the periphery and all the different technologies leading to, we call it tissue engineering,
Hmm.
um, I thought it was worth a go. So this is where I am and what I do.
Really cool. And we'll talk about, how all of this works in a moment. I think what's important for maybe the listeners who might never have heard about, bioprinting, um, there's, some terminology I'd like to go through with you. So,
Yeah.
how do you define bioprinting? I think when I check on your website, you also mentioned biofabrication. So how it relates to the printing aspect, and then the broader, let's say term that you also use, I think, with Cellbricks, which is tissue therapeutics. So delivering tissues as therapies. I think it would be great if we could, define these three terms.
Absolutely. Well, bioprinting and bioprinter. So bioprinter is to me an instrument, it's a tool. And
Yeah.
bioprinting is the function of creating cellular constructs, three-dimensional cellular constructs. There's many different types of bioprinting technologies out there, from things like extrusion, where it's like a syringe that squirts very fine streams of cells, or biomaterials into a dish or a plate. Then there's other technologies like we have, which is light-based, where
Yeah.
there's a light-sensitive cross-linker within the bioink. Light shines into the bioink, and where the light shines, you harden. So the idea for bioprinting or 3D bioprinting is to create 3D cell constructs based around some form of biomaterial type scaffold.
Yeah.
And then from there, that is one part of the process, which is biofabrication. So biofabrication doesn't just involve the bioprinter, it also involves the cells that you're going to use. So these cells come from either a patient, so they are primary cells from the patient itself, or they are coming from a stem cell source, and you have to expand them to a certain quality and a certain standard. You also have to formulate the biomaterial. So the biomaterial, or bioink we'll call it, is a composition of chemicals and proteins, things like collagens and hyaluronic
Yeah.
acids. And this needs to be very carefully formulated as well so you get repeatable printing. So all these pieces of that puzzle is the process of biofabrication. And biofabrication can be used to create mini tissues for drug discovery testing. And as you're probably aware, both in Europe and the US, there's a huge drive to move away from animal testing in drug
Mhm.
discovery.
Yeah.
And so you can get human cells and you can create multicellular constructs, potentially even with some type of perfusion or vascular system within these microtissues to try and mimic the real situation in the human body and try and get proper or more predictive data. So this is biofabrication, but I think if you were to really ask, the holy grail or the idea behind 3D printing is, well, what if we could actually print an organ, regenerate organs? So anything within the body, and we could start with things that may be relatively simple to print, such as bone or teeth, and they are
Yeah.
now commercially available. And that's really more a function of 3D printing than bioprinting, but there's maybe
Yeah.
cells. But then you say, well, for people who have um, transplantation need, we can now take cells from those patients and we can generate stem cell lines unique to that patient. We can expand them. We can turn those cells into the organs, so liver cells or kidney cells. So the next step would be then putting all these together with the patient's own cells and creating a new kidney for them, which, as you know, there's a huge demand, a huge shortage
Yeah.
of
Yeah.
organs
Mhm.
for transplantation. So that I think is I think it's unfair to call it the moonshot, probably more like the Mars shot, but that is what
Yeah.
we all
Mhm.
work towards. And that is the goal that brought me to Cellbricks. And that is what we mean by tissue therapeutics. It is a therapeutic piece of tissue or eventually organ that would be transplanted or implanted into the patient to recover loss.
Mhm. Understood. And so I understand as you mentioned, the Mars shot, which would be like to really print a whole, like, fully functioning organ that's probably like, some decades away. But I'm also wondering, like, what are some of the, let's say, very concrete medical needs that where we can have like, applications in the near term if,
Yeah,
we manage to, still make progress in that regard.
absolutely. So two concrete examples, one from Cellbricks and then one from one of the other companies within the space. So Cellbricks, we are a tissue therapeutics company. We don't sell our bioprinters. We don't
Yeah.
build disease models. Our only goal is to try and recover tissue loss. And our first target is, we can maybe talk more later, is wound. So it is soft tissue that we're creating, vascularized soft tissue that can be printed into the shape of a deep wound. So a wound that wouldn't heal. So it can be anything from a terrible trauma through to maybe a ulcer, like a diabetic ulcer, to a terrible burn that's down to bone or cartilage. We
Yeah.
can take the patient's cells, we can print a piece of their tissue effectively, and it's almost like a plug. It goes into the patient and effectively heals them. And we are at the stage now where we are just transferring that to the manufacturing, the clinical manufacturing organization we're going to work with. Because I've talked at the moment very research side of tissue engineering,
Yeah.
and now in the second wave, we now have to start understanding the clinical. How do you get it into humans? How do you do the safety testing? How do you manufacture it? And so our wound tissues will now be in that safety testing stage. And our aim is to put this into people in 2028. So that's our
Mhm.
near-term goal. Another where maybe more organ function is replacement of beta cells. So obviously diabetes, there's
Yeah.
huge drive in that whole area at the moment. Um, and it's relatively straightforward to get human beta cells to generate insulin. And you don't need a lot of those cells.
Mhm.
Something like a liver, you need billions of cells to
Yeah.
recover liver function. For beta cells, not so many. So companies like Aspect Bioscience in Canada are creating small constructs with beta cells in them that would release insulin, and they're working with their pharma partners. And again, they're in the safety stage and they're looking to put that. So those are concrete examples of tissue engineering that in the next few years has a good chance will be in the clinic
Mhm.
and
Mhm.
potentially will be in patients having some therapeutic effect. Something like a full liver or a kidney, as you say, is very far away. But we see even for Cellbricks, our other program is liver, and we're creating what we'd call a liver graft, which would be a small portion of the
Mhm.
patient's liver that would recover the liver function or recover some of the enzymes that the liver should be releasing, particularly for some of these horrific inherited metabolic diseases that children suffer.
Mhm. Yeah, I was also, I think also it makes sense from when you think about the liver, I think it's one of the organs in the body that regenerates very well, probably, with the guts. So that, that's probably well suited. I was also thinking when when you talked about, wound, the applications in the, for wound management. There's a company here in Zurich actually, that's led by Daniela Marino. I forgot the name of the company, but they're doing something with the skin, right?
Yeah.
Um,
So there's another good example, you're right, is skin. And skin, particularly companies like L'Oreal, have been
yeah.
leaders in the field where they've been able to, using bioprinters, generate multicellular skin models that have
Mhm.
hair follicles and sweat glands. And I mean, obviously, they have their primary role is for the testing of materials, but clearly that also has a role potentially in therapeutics.
Yeah.
But this then
Mm.
brings you to the second stage, which is really where, if you say, our barriers are now at that next stage. So the bioprinters work well. The biomaterials, we're able to formulate them well. Vasculature, we can talk about how well we can do vasculature, but in small scale, we can create a sufficient microvessels that will allow the tissue to live, to perfuse, and not to die.
Mhm.
But now you come to actually the side of how do you make money out of these? Because clearly we're all getting investors' money, and the investor would like a return or company to be acquired. And for there, suddenly it's a whole new world, and that is the world of how do you manufacture these repeatably? How do you make lots of them? Where are those? Where are they based? Are they based at hospitals? Or are they based at a
Mhm.
central production facility? What do the regulators actually want you to do before you can put it into humans? And this is the place where we're at today, where there's a lot of discussion going on, and even down to, so we can make it all, but would it be too expensive? Would anyone, any insurance company or government actually pay for the construct? So for a
Mhm.
liver for transplant, I think we could charge any price we wanted. And it's not that we're looking to make lots of money, it's just that these things are going to be incredibly expensive today with some of the technology limitations. And that's coming back to the Mars shot, that we can see how to do many of these things, but scaling or making it commercially viable,
Mhm.
we're right at the edge. And I think there's multiple companies out there, and we all know the same challenges. And at the moment, I feel it's not actually competition. We work, there's more than enough space for us all. But it's a translation away from, there's a lot of academic groups who are still building the building blocks, the early
Yeah.
building blocks, but it is up to the therapeutic companies like ourselves to now push the commercial part, the regulatory part, so that one day, and hopefully very soon for us, the European and the US regulators, they have a book and we just follow the book. We know how to grow these things, we know how to build them, we know how to
Mhm.
manufacture,
Mhm.
and we have a product that could go in the market. And it sounds relatively straightforward, but no one's done it before. So it's
Yeah.
we all have to work together to solve these. And this is where wave two or even wave three of bioprinting, that's really where we are at the moment.
Yeah, I just remembered the name of the company. So from Daniela Merino, it's called Cutis. So they're engineering skin to place for, I think it's mostly for
Yeah, yeah.
burnt wounds. Exactly. So something probably also to have a look at. And to your last point, I was asked, I wanted to ask you the question on, what is actually the level of evidence that we have that, bioprinted tissues or biofabricated tissues can actually heal. And I guess, when you look at the work, I think in the case of like the cuties there, I'm not sure if they're doing like this, following a similar process as you guys do, but there are probably in certain indications already some signs that this is actually working. I don't know if you wanted to elaborate on that.
I think, and this is where there's the partnership between the research groups, the academic groups,
Yeah.
and the companies. The companies, it's very difficult for us to get paid to do basic research. We need to apply very quickly, and our runways from the investors is always short. But if you look based on the evidence that's coming through numerous animal studies in every tissue or organ you can think of, even down to really complex things like kidneys and livers, there's evidence that you can get longer term survival of tissues and they will do what they're supposed to do. So for liver, as example, at the moment, one of the hot spaces is, uh, could you infuse liver cells into a patient who has some liver failure, so an acute liver failure? And there's a number of companies that have developed the ability to create spheroids or organoids, so groups of liver cells. They coat them so that the liver cells are not attacked by the immune system. They infuse them into the bloodstream, and these liver
Yeah.
cells live. The challenge is that there's nowhere good for them to embed and then to thrive. So it's a numbers game. You put a certain amount in, a lot of them get destroyed very quickly as they pass
Yeah.
through the circulatory system. Some of them lodge in the spleen, or some of them lodge in the liver, and they recover some of the patient's liver function. But it's short term because they're not embedding into a
Yeah.
cellular space that encourages their growth. So then you
Mm.
say, well, the next level is, why not create that cellular space for them by bioprinting, putting blood vessels in and going from there. And the microcapillary, or the small scale blood system, I think again has been shown to work in vivo. In humans, we still need to do more work there, but as I say, a number of us are working towards that. Where it then becomes challenges. So if you want to scale up further, you could imagine you're going to need larger blood vessels. So the microcapillaries keep small to medium-sized constructs alive. When you start going to bigger things, you need good blood flow. And from there,
Mhm.
suddenly this is a whole new engineering problem, and we're still trying to solve those problems. There are some groups who've done some excellent work on that. But how do you even connect your bioprinted construct? How do you have a connector between your large blood vessels and your bioprinted construct and the patient? A surgeon has to be able to suture those. So you have to create something that is suturable, but also that will be biocompatible. And this is where all these things I was talking about that are within that biofabrication sphere are still all cutting edge. We're still developing all the
Yeah.
time new biomaterials, different types of cells. How do you make it immune silent? Can we engineer the stem cells so that it's hidden from the patient? Or could we work a way where we can actually regenerate the patient's cells so that then we can put it back into the patient? And there are many companies out there that are each tackling different pieces of this puzzle. And where Cellbricks is, we are, I guess, an integrator of some of these puzzle
Yeah.
pieces.
Mhm. Really cool. I wanted to ask you about, what is exactly the biofabrication process that you follow, and I think it would also be, a good way to understand as well, like what's currently feasible and where we currently have challenges, which would have been my follow up questions. But I think it would be great to dive a bit into the process that you guys follow and
Yeah.
how the technology works.
So because there are so many ways to do this, there's no one way is best. So
Mm.
Cellbricks, as I said earlier, we use what is called a light-based bioprinting technique as opposed to extrusion.
Yeah.
And I don't think it's not a function of one is better than the other. They all have their own areas that they're particularly suited to. And a light-based system, um, say effectively you create a bioink, which has a light sensitive reagent in it, that where the light hits, it hardens the bioink to become a jelly. And you can control the stiffness and the porosity of that jelly, so it can be incredibly soft to being really quite hard.
Mm.
Cells like soft things. They don't like being in hard things. But on the other point, a surgeon is not going to be able to pick up an incredibly soft piece of jelly
Yeah.
and do anything with it. So you have to work out that idea where there may be soft parts and hard parts so that you have something that you can use. So our light-based system can create a layer which can be anything from a few microns to a few hundreds of microns thick. And the layer size is dependent on the intensity and the length of the light flash, but the light flash can be a few seconds. So you can create a layer every few seconds by a flash. So our system is very good at creating large structures quickly. And we can, by changing the bioink we're using, which has different cells in it, we can add different cell features. Or we could add a harder outer surface to our construct so somebody can hold it. So that our system creates large constructs quickly, and it's also very gentle on the cells, which is another important part. Sometimes there are certain cells that don't like going through the extrusion system, which
Yeah.
is in a needle. Cells being pushed through a needle sometimes isn't good for certain cells. So we know we can get sufficient resolution. We're not claiming incredible resolution, but for us, it's not necessary. We can build these large constructs. So for the wound program, our ultimate goal actually is to build breast for reconstruction post mastectomy. And there we're talking of tissue constructs that are between 200 ml and 400 ml in size. And we
Yeah.
need to be able to print them within an hour so that we
Wow.
can do the full print, and then we can make sure that fluid is flowing through them so that the cells mature, that the construct grows, and that you don't get cell death within your construct. So that's our key feature. Um, we're also very good at Cellbricks. The other strength we have is in formulating these bioinks. So there's core materials, so there's collagens and other core starting points, but then there's different types of extracellular matrix proteins that are unique to different parts of your organ, and also maybe growth factors or other proteins that might be important, not just to keep the cells happy today, but actually to maintain their functionality over the
Yeah.
long term. And so being able to fine tune the bioink to make sure it will do what it's supposed to do. Is it supposed to degrade? So is the host supposed to degrade it? So in our wound product, we would like our material to be replaced by the patient's own cells
Yeah.
and extracellular matrix. In a liver, maybe less so. We'd probably like the structure to be maintained for a long period of time. So the formulating the biomaterials is also an important part of the process. And then designing the construct. So having software or design function that allows you to figure where to place the cells, in what orientation, at what concentration, how close or how far away from vascular or tubes that would allow fluid to flow through to feed those cells. That's another important part of the process.
So, and you master all of these three aspects. And I think, I mean,
Yeah.
when you see what you're showcasing on the website, it's very interesting. Like I spent some time looking at how the software looks like. So you can really design in 3 dimension how, yeah, ultimately the tissue will look like in the end. And then I guess another part of the challenge is once you've kind of like figured out these three aspects, so what you want to design, once you've got the printing part right, and once you've used also the correct bioink, there's this question that you alluded to when you said like a surgeon cannot use something that's too soft or disaggregates. It's a bit like, how do you transfer the tissue, sort of like graft and what you've printed from the printing lab to the clinical setting. Because ultimately, if you think you like, if we take the scenario, we operate in a hospital where, hospitals have their own labs. I don't know if in the future they will have, their own 3D printing labs, but that's a way to, make the connection very quick and avoid, maybe transportation delays and so on.
Yeah. That is an excellent question, and that is one of the parts that is a problem to be solved. So
Yeah.
as I said just before the call, I've just returned from our labs in Cambridge, Massachusetts. And there we're working with an organization, a company that are going to help us industrialize our process. So turn it into GMP, prove that we can prepare all the different parts to a clinical grade so that we can go into
Hmm.
humans. But as you say, the next is a logistics puzzle. So I've already talked about the regulatory puzzle that we're all
Yeah.
working towards. What do the regulators expect from a tissue therapeutic? And in the regulatory part, most new therapeutics, first of all, go into a phase 1 clinical study, which is into healthy volunteers, and you look for safety. Our tissue constructs will not be coming out the patient easily once they're embedded into them, because they will integrate into the host's
Yeah.
vasculature.
Yeah. Mhm.
So we're already doing a phase 1 and phase 2 study was what we would need to do. So that requires a different set of considerations from the regulator. But the piece in the middle, and this is also why we are talking Cambridge to the big hospitals there and the clinicians, it's not just getting it to the clinicians. How long can it survive post-print before it goes into the human? What is the process that surgeons would use? Well, they've not done this before, so we have to work with them to tell them, how does this arrive in the operating theatre? What do they do? What do they have to do? And clearly, if it's too complex or they touch it and it breaks,
Yeah.
it'll never be accepted. So it'll be a great research project with an interesting science paper, but that's not what
Mhm.
we get paid
Clinical
to do.
application. Mm.
So back to the word biofabrication. So I've talked about things at the front. At the back, there is this, we'll call it transportation system. And this is also something that we have built. So again, one of the things that attracted me to Cellbricks was we've talked about the biomaterial and the cells and the design. But the bioprinter itself, I don't believe you can call yourself a tissue therapeutics company unless you can also control your ability to manufacture the tissues or the organs. And as we discover that the bioprinting system needs something else, or that we have to create this transportation system that allows fluid to flow through the construct so it can be
Yeah.
maintained functionally, so a perfusion system. So we've built that as well. And this, thankfully, there's a great team of hardware and software engineers, as well as the biologists and the chemists and the tissue engineers at Cellbricks. So I am lucky enough to have an engineering team that have built this transportation system that would take it from the
Mhm.
time of bioprint to the time of entry into the patient. So you're absolutely right. This is yet another one of those challenges that we're all working to try and solve to turn it from a cool research project to reality.
Pretty cool. We'll have, I think one of the next recordings I've planned is with Lisa Anderson from Paragonics Technologies. So it's a company that's providing ways to transfer organs, like proper human organs,
Yeah.
and, keep them. Because normally they just put them in ice. That's what I've learned. It's very like a basic way of, transferring one organ from one place to another. Just put them in ice with the risk of, freezing them, and then you have, functional damages that you cannot recover. Um, so yeah, there are probably some interesting insights from that future conversation that relate to what we just shared.
I mean, obviously, I guess in a lot of this, it would be great to think you have to create lots of really cool new technologies, but
Yeah.
clearly we just need to get to the endpoint. So being pragmatic is often the best approach to start with. Unfortunately for us, just putting our construct on ice or
Yeah.
a cool chain just doesn't work because it is still an artificial system. We do
Yeah.
need to drive fluids through it. So there is there's a mechanical process that we need to add into our constructs. So we built this as part of our biofabrication process.
Really cool. And so for now, the fabrication and the GMP, grade production site, that's in the US, as you said, because there are two. For the moment, you're located in two places, right? There is a
Yes.
Berlin office, a Berlin location, and a Boston location.
Absolutely. So we came out of Berlin University, and the full research team is in Berlin.
Yeah.
The challenge, though, is um, because what we're doing is not standard, it is
Mhm.
finding other partners that can help with what we just talked about, these other new steps. And some of them is not necessarily new, it's just being able to interpret regulatory rules to suit. Because if we get it wrong and have to go back, that could, you know, be
Yeah, yeah.
very bad
Mhm.
for our company. And what we found that our partners that we've identified both in hospitals and also in the manufacturing come with a set of unique knowledge that gives them a step up for us today. So we are both in the Europe and the US, but our manufacturing for our first product is US based.
It's US based. You mentioned that, you were already into phase one and phase two clinical trials for the therapeutic programs that you guys are developing. Um,
Just no, we will be at the moment. We're still
sorry.
in the no. I was just say, so yeah. Just to be very clear, we're at the safety stage. So
Yeah.
the
Okay.
preclinical safety stage.
Preclinical.
I would
Okay.
like us to start the clinical trial at the start of 2028. And
Thank
there,
you.
what
Okay.
I was saying was that the clinical study has to be designed differently because it would be a phase 1, 2 combined study because
Yeah.
we we just can't put our constructs into healthy
And bring it
volunteers.
out. Okay. Yeah. Sorry, I got a bit ahead of myself. Because my question was, how far are we from, seeing what you guys are working on, used in clinical trials, and
Yeah,
you answered the question, and then potentially, in actually in, clinical application.
I mean that is our goal, and we're not alone. As I said, there are companies that are
Hmm.
working on printing liver patches, on islet patches, on kidneys. There are heart is a hard, but there are people also in the heart space. And then lungs, I mean, United Therapeutics with their lung program, just incredible the things they're doing too. So we're all racing forward, all, I think, finding similar challenges. And so some of the most valuable things are opportunities to meet up with we don't see them as competitors. We're
Hmm.
all in the race. As I say, there's more than enough space for us all, but it's trying to share where at all possible experiences that could be applied across multiple organs or multiple systems that help us all move forward.
Yeah, and it's such an incredible challenge. I mean, first, it's very impressive to see how far we've gone with the technology and how, what remains to be achieved in terms of technological progress. But when I think it was also quite revealing for me when you said also on the regulatory side, we need to rethink the way we do phase one trials, potentially phase two, and, mix them together. Like, it's not just like, I mean, as if let's say, the technological development was not like difficult enough. There's a whole, change that needs to happen also from a regulatory perspective until, eventually you guys, make it into a, clinical use, which I really hope, happens down the line. But I think it's incredible because it's really opening up a new way of, treating people at the end. And it's, yeah, building on, many things we've seen over the past few years. I think it's a very, very exciting, field to be involved in.
And this comes back to, I think, the question you asked at the start, why am I why am I here? It's so there's no therapeutic area that's easy. You may say small molecules are better understood, but there's still challenges. But the ability or the opportunity to be involved in that, someone never having done it before, and helping push the whole field forward, being part of that is yeah, not only incredibly exciting, but I feel incredibly lucky to have the chance. And I'm also incredibly lucky because a we're not a lot at the moment, but there's a growing number of, I guess, what we would call tissue engineers that are coming from a number of good groups across the world. And we've been very lucky to attract a really talented group of people in Berlin, which allows us to try the things that we've been talking about. So I should not forget that it's not just the hardware and the software and the chemistry
Yeah.
and the biology,
Mhm.
it's the people too. And there's a growing number of us that truly believe that this is going to happen. And the only way it is going to happen is if we keep pushing hard.
Really cool. So there's, keeping an eye on the time as well, and there's still, a couple of recurring questions I wanted to ask you,
Please.
Simon. So the first one is, what resources would you recommend our listeners to look into, to know more about the field in which you work? It can be, books, publications, websites, anything.
Yeah, it's an interesting question. And I say interesting because I've talked about the waves of bioprinting. And actually, I think one of the problems with the first wave was that it was overhyped,
Mm.
that a lot of work was done. It was early days. People have got a bioprinter. What do I do with it? A lot of material was generated. A lot of publications were generated, but it didn't really have much in the way of application. It was I printed a cell line. Now I've printed another
Yeah.
cell line, or I put two things together. Now in the second wave, there are some incredibly good groups that publish, but because it's such a broad space, from the hardware software through to the materials, through to just generating cells, trying to find something that actually combines it all is not so easy. Um, there are a number of organizations that have that I guess are hubs for us. I mean, one in the US, the Advanced Regenerative Manufacturing Institute, they have been funded by the US government to be a center, a hub where everyone can come together. They share a lot of information and non competitive information. They organize a lot of events. And they, here in Europe, is maybe a little more fragmented. So the Dutch are very good and have their section. In the UK, Manchester is very good, and there's a centre there, the Royce. Berlin, or we have pieces. You've said there's a number of good groups in Switzerland.
Mhm.
It's more fragmented. So I think there's a lot of material out there. I think the challenge is always if you're looking for a high level or
Yeah.
what
Yeah.
is possible, that is more difficult to pick through at the moment because just as an effect of it being an early field, people often have to extrapolate what is possible without it actually being possible so they can justify their funding. So what is
Yeah.
real to what is a wish. So I think one of the best resources that I know is this Advanced Regenerative Manufacturing Institute, who are incredibly open with what they share. And we're a member of them too. I
Yeah.
think almost everyone is in the field so that we can share information.
Really cool. So I'll put all the links in the show notes. It's always a bit like maybe early for you to answer the next question because it's one where I'm asking, the guest about an anecdotes of, about their work that made them realize the impact that you were having on people's lives. And that we mentioned before that in your case, the clinical trials are, coming on the horizon, but maybe there are like maybe interactions you've had with doctors or, with physicians, surgeons, people in the healthcare ecosystem that might already, provide some interesting anecdotes you wanted to share.
I think very simply, it is that interaction with the clinicians that makes it real. So our wound product, our wound graft, in some cases is not incredibly complex. It's not a liver or a kidney. It
Mhm.
still is very
Yeah.
difficult for us to make. But there was a tendency for some people to call it the low-hanging fruit because yeah, it doesn't have a function. It's soft tissue. It doesn't do liver function or kidney function. But we then had to understand what was the real clinical need. and going to clinicians across Europe and the US and actually talking to them about the patients. And every one of them said, well, this material, I have a patient upstairs who's had a wound that hasn't healed for 4 years. And at some point, they're going to lose their leg.
Mhm.
And what I hadn't realized that your survival rate, your 5 year survival rate post limb amputations, 20%. So your chance of survival just for that is so low. And
Yeah.
the effect that we thought if we could stop, and it's the infection in these wounds. The wounds continue to become infected because they're
Yeah.
open sores. And it's not just the skin graft doesn't do it because there's this big defect. This I've had clinicians saying, forget the clinical study. My patient would sign anything. If
Mhm.
they would give you their cells, they would sign anything for us to try it because we've tried everything else on the market and they know they're coming to the end and that
Yeah.
very soon they'll lose their leg and not only quality of life. I can't imagine how bad that is, but actually their survival rate is so low. So when you having been in the therapeutic space a long time, I've seen this in other with cancer drugs or other, but it's really hard for some of my team who are young researchers to hear these clinicians saying
Yeah.
how desperate their patients really were. And then the question, well, do you think we could? I mean, unfortunately, there's no way we can just skip all the safety studies,
Yeah. Yeah.
but the feeling is there.
Mhm. Oh, thanks for sharing. If you would recommend, another fellow healthcare innovator as a potential, guest for the show, who would that be, and why would you recommend, her or him?
Yeah, well, how interesting, because I did think of Eric, because Eric was a visionary in that he brought together, first of all, he democratized bioprinting.
Yeah.
And I am going to focus on bioprinting because or biofabrication, tissue engineering, because I do think it is something that is coming. And there's so many interesting people that are pushing that wall further and further away. And Eric did that. But someone else who's doing it is Carolyn Collins at Dimensions Bio. And they have a very different way of doing bioprinting. They've got a very different philosophy for how they're trying to solve the problem. But they have really, again, pushed boundaries and they have managed to get a product through FDA approval. So
Yeah.
they've done what we dream of or we're trying to do at the moment. So that would be somebody else that I would think to get a different perspective on this space. And maybe your listeners and viewers will have had more than enough of the bioprinting world, but to me, you could never get enough.
Yeah, yeah. No, I would agree. So I'd also be, super interested to hear their perspective. Well, Simon, thank you so much. You've been, very generous with your time, and I'm really happy that we, had that conversation. I think it was really eye-opening for me, and it was also, from a very, let's say, like let's say, honest and, I would say, grounded, perspective, because we can talk about, printing organs and so on, but this is like still very much, far away. And I guess when you talked about the fact that bioprinting was overhyped, I think there was also a bit this sort of effect, right? When the first bioprinters came out or when the technology got into the media, it created a lot of like fantasy and there are, maybe some crazy ideas, and actually was not really linked to what the technology is actually, capable of doing. And I think in our conversation today, you've, articulated very well where we're standing, what we can expect, in the short term, and what are the challenges as well. And I think it was really, really interesting. So thank you so much.
Thanks, Matt. I will just say I am very passionate about this space. So when you're talking about resources, I'm also always very happy to talk about it. And I do think this second wave that we're in now is a much more grounded and realistic wave, where we are now thinking of those further steps that will get us into the clinic. So I don't doubt within the next 5 years there will be multiple, not organs, but certainly multiple bioprinted tissues that will be having a therapeutic effect and benefit to our patients. So give us a few more years and we will be there.
I'm looking forward to that. Thank you so much, Simon.
Well, thank you too. Thank you.
Thanks for watching the episode. I hope you enjoyed it. You can subscribe by clicking on the podcast channel at the top right and watch another episode here at the bottom.